Today we're going to start talking about Mendelian genetics. Have you ever looked at your family and thought, well, I have so-and-so's hair and so-and-so's nose? We do it all the time in our family. I have a lot of redheads in my family, and as you might know, I have very dark hair. So I have an interesting combination in my family because I am part Native American and my mom is a redhead with green eyes of English descent. So it's really fun to start talking about inheritance and how things are passed down. So today we're going to start looking at that. And of course, our genetics have to do with all the chromosomes that we talked about. And here is our karyotype with all those chromosomes laid out. So let's talk about some of the terminology of genetics, just so that, again, we're on the same page. So we've mentioned a gene before. It's a stretch of DNA that codes for a protein, and it is found on a chromosome. So the locus, or plural being loci, is the location of the gene on that chromosome. And then we also use the term trait, which that one should be pretty straightforward. That's the feature you are studying. So for example, eye color, or because we're talking about Mendel, pea plant height. And you'll see how those are connected in a minute. Or it could be hair color. Okay, so let's say that the trait that we are interested in is your hairline. I don't know if you ever knew that this was a genetic trait, but this style of hairline that comes like this is called a widow's peak. And some have a straight hairline that does not have that peak. Now I have to tell you a funny story because I do have a widow's peak. However, since I went skating with my daughter one time and my skateboard hit sand and stopped, and I did not, this is a lesson in always wear your helmet. I was not wearing a helmet and I fell and landed pretty much right here. And other than the concussion that I had to deal with, I lost all the hair on my widow's peak. And so I looked like this, despite the fact that I have a widow's peak. Now, it is sparsely growing back in, but I will say I miss my widow's peak. Okay, so let's say this is you. And you have your one chromosome from your mom and your one chromosome from your dad. And for our example, we are going to say that we're still looking at the trait, the hairline gene for widow's peak, and this is its locus right here on your chromosome. Now, when we use the term allele, that means different versions of the gene. So the two alleles for us would be the presence of a widow peak or the absence of a widow peak, or another way to say that would be a straight hairline. And those are the two different what we call phenotypes, and I'm going to get to that in a minute. But the allele is the different versions of the gene. They code for two different things. So if you have two different versions of the gene, then you are what we call heterozygous. Like for instance, if you got a widow's peak gene from your mom and a straight hairline gene from your dad, then you are heterozygous. And that term, heterozygous, just means one of each allele. Now, if you had two of the same version, you are what we call homozygous. So in other words, if you got the widow's peak gene from your mom and the widow's peak gene from your dad, you would be homozygous for the widow's peak trait. Okay, so I mentioned this word, I mentioned it a little bit early. So your phenotype is how you look, or your chemistry or your behavior. So in other words, when you have that straight hairline, that's a phenotype. But the phenotype is determined by the gene. And so the genotype is the individual collection of genes that you have that produce the phenotype. Does that make sense? So your genotype is your bunch of genes, and they determine your phenotype, which is basically how those genes look when they're expressed. So our hairline gene codes for a widow peak here, and our hairline gene coding for a straight hairline here. Those two are the genotypes, so we would say you're a heterozygous genotype, but your phenotype would be a widow's peak. So you would look like you had a widow's peak. And here's why. When we have different alleles, one of those alleles is going to be what we call a dominant allele. And that's the one that is expressed. That's the one that will give you your phenotype. The other allele is the recessive allele. And that's a gene that's either going to be masked or unseen. So in our example here, our gene for straight hairline is a recessive trait, and a recessive allele, and the widow's peak gene is a dominant allele. So what you're going to look like has to do with the dominant allele. So in other words, the phenotype is based on the dominant allele. So here is what it looks like. So here again we have our hairline gene coding for a widow's peak and our hairline gene coding for a straight hairline. And remember, this is heterozygous because we have two different alleles. And because the widow's peak is a dominant allele, then this is what we're going to look like. Leonardo DiCaprio has it. See how these famous people have this widow's peak. Okay, so when we're going to write these things down, how do we denote them when we're trying to describe these to people? So when we do notation in genetics, we use the first letter of the dominant allele to talk about the gene. So in other words, our trait is our hairline gene. The dominant allele we said was the widow's peak, whereas the recessive allele is the straight hairline. Well, according to this notation, we take the first letter of the dominant allele, which is the W, and we would write that as a capital letter, so a capital W. And by default, the recessive allele, which is the straight hairline, gets the lowercase letter, so a lowercase w. So when we are talking about the genotype, then this is how we would denote it. Our homozygous dominant would have two capital W's because we have two dominant alleles. Because remember, homozygous means two of the same. Dominant, in our case, is the widow's peak, so we have two capital W's. And our phenotype for that particular genotype is going to be the presence of a widow's peak. Now, our earlier demonstration that we talked about was heterozygous, right? We got the widow's peak from your mom and the straight hairline from your dad. And we represent that by, we have the widow's peak from mom, that's our dominant, and the recessive allele from dad, that is the small lowercase w, which indicates the recessive version of the widow's peak gene, which is a straight hairline. And heterozygous, of course, meaning two different alleles. And because we have a dominant allele in there, the phenotype will still be a widow's peak. Okay, then our homozygous recessive. What if we got straight hairlines from both mom and dad? Well, homozygous, meaning two of the same allele, in this case, both recessive alleles, we still indicate that by the lowercase w because that is the recessive version of the widow's peak gene. Okay, and so this is the only version in which you get a straight hairline because of the absence of the dominant gene the recessive gene can then be unmasked or demonstrated. So homozygous, both of the same alleles, but both recessive, in this case, is going to give you a straight hairline phenotype. Okay, so if we are going to review these terms, let's take a look at this. So here we have our chromosome, and on our chromosome we have 3 different genes in 3 different locations. So these are our gene loci. So we have gene P right here, and it looks like we have a dominant P and a dominant P because they're both denoted with a capital P. So I know that that is a dominant gene. Now because they are both dominant, that is a homozygous dominant allele. Our second gene at our second gene loci is we have a lowercase a and a lowercase a. That indicates that we have 2 recessive alleles. So we would say that this is homozygous for the recessive allele. Our third gene at this third gene locus has a capital B and a lowercase b. And because of that, we would say it is heterozygous for this B gene, and we would denote that by a capital B and a lowercase b. Now, one thing I don't want you to get hung up on is this says recessive alleles, which these right here are, and this is a dominant allele here and here. However, it is not the case that recessives are always on one chromatid and the other is on the dominant, because remember we had random assortment. So I just didn't want you to get hung up on that. Okay, so that's our review of our terminology. So this is Gregor Mendel, and he deserves so much credit having to do with genetics. I want you to keep in mind that all of his research was done before they knew anything about DNA. So at the time, everybody thought that you got some kind of information from your mom and some from your dad, and that you were a blend of the two, which means that everybody would start looking more and more alike. And he did meticulous research using pea plants and statistics and probability to come up with a different theory, and his was the particulate theory of inheritance. And so since he didn't know about DNA yet, those particulates are what we now call genes. He knew that there were very distinct particles that we passed on to the next generation based on his studies. So I just want you to keep in mind the lay of the land when he was doing his research, because the other thing is people didn't use statistics at the time during their experiments, but he had a math background and applied that to his study of inheritance. So he used pea plants when he was looking at these inherited traits. Why pea plants? Well, one is that they're easy to breed, they produce a lot of offspring, and peas have many visible traits that would make them easy to distinguish between each other. Okay, so let's get familiar with sexual reproduction in plants. We've mentioned it before, but let's talk about the different parts that are involved. For instance, all of our sexual reproduction in flowering plants happens at the flower. So the flower is the sexual organ, or holds the sexual organs. So the male portion of the plant is this stamen right here. In the very top, that orange part, is called an anther, and that's where the sperm is produced. The female portion of the flower is this stigma area that leads down to the ovary with ovules right here. So what happens is a sperm is produced and as part of pollen, and as it flies through the air, then it will land on a plant and a flower at the stigma. And the stigma has a little bit of stickiness to it, so it will hold on to the pollen. And within that pollen grain is a particular cell that will then grow a tube right down here to the ovary, and that tube is meant for the sperm to swim down and fertilize the eggs, which are of course in the ovaries. So this is just a cutaway view. So here you can see the stamen with the anthers, and then this is the stigma that underneath has the eggs in the ovaries. So again, during pollination, which is the process of the pollen sticking and then the sperm growing down through the stigma to where it reaches the egg and can fertilize the egg. Of course, when it does, each one of these is a baby pea. It comes from the fertilization. And each one of these can then be planted and grown to adult plants with a variety of traits that we can then analyze. So when we're talking about flowering plants, there are some interesting phenomena. So unlike animals, plants can self-pollinate. So in other words, pollen from one flower fertilizes the egg in that same flower, and it's perfectly healthy. And so we denote that by this symbol here with an X that has a circle around it. When pollen from one flower fertilizes the egg from another flower, that's called cross-pollination, and we denote that simply with an X. Okay, so here is basically the pollinating of peas, and so we just want to get some terminology. So we start with the pea generation, and this is going to be like the parental generation. So these are the parent plants up here. And in this particular experiment, we have T for tall, and then of course the recessive allele for the short is the lowercase t, and what you can note about these two is that they are homozygous. So then we have the parental gametes. What are they contributing to the cross-pollination? Well, obviously, either that is produced from this pea generation is going to be the tall, and from this one it's going to be a short gene. And that gives us what we call the F1 generation plant. And F just stood for filial. So sons and daughters is what that means, but we can kind of relate it to familial in English. So the filial generation, the F1 generation, so the first sons and daughters. And this cross gave us tall plants but are heterozygous. And from there, then we can decide what kind of gametes would these particular F1 generation plants produce. And so we have the sperm over here, sperm options. So this plant could produce a tall or short sperm. Our egg options is it could produce a tall or short egg. So the possibilities for the F2 generation are in this right here, which we call a Punnett Square. Okay, so when Mendel started his experiments, the first thing he needed was what we call true breeding parents, or purebred parent, for that parental generation. And that means that the type of egg and sperm that they can produce only produce one type of offspring. So essentially, when we have a tall pea plant that has been self-pollinated over and over and over generations, eventually it can only produce tall plants because we have selected for that, and we come out with a homozygous tall plant. Same thing for our purebred short pea plant. It has been self-pollinated for generations to where it only produces short plants, and it is homozygous for short. Now, our first step in this is that we have cross-pollinated 2 purebred plants. Okay, so we have our gametes that we talked about a minute ago. We get a tall gene from our tall plant and a short gene from our short plant, so we cross-pollinate these two. And by the way, this was done by hand by Mendel. He collected pollen from one plant and he brushed it on to the style of the other plant. So very tedious work. And so when he did, we wound up with the F1 generation that we talked about with a heterozygous tall plant. So if we want to cross a heterozygous tall plant with itself, so here we go for self-pollination, then what eggs meet up with what sperm? And this is where we use the Punnett Square. So again, the sperm from this particular plant, our F1 generation, the two possibilities are tall and short. And the egg, same thing, tall and short. And so then what we do is we say, well, if I have one gene from here and one gene from here, I have a homozygous tall. If I take one gene from here and one gene from here, I have a heterozygous tall plant. Over here, again, one tall, one short, a heterozygous tall. And then over here, I have 2 recessive alleles from here and here that produce a homozygous short. So we use this Punnett Square to predict what could happen. So when we're looking at a cross that involves only one trait, and in this case what we're looking at is whether it's tall or short, and that's all we're looking at, we call that a monohybrid cross because it's one trait. And how do we know that you'll get a 3-to-1 phenotypic ratio? Well, that is what we just counted, right? So we know that a homozygous tall is going to give us one tall plant. So here we're going to put in here tall versus short. You know I can't write with this thing. So far we have one tall. A heterozygous tall plant will also look tall. Again, a heterozygous tall plant is going to look tall. And then a homozygous short will look short. So when we have a self-pollination of a heterozygous tall plant, then the phenotypic ratio of the offspring from that will be a 3-to-1, 3 being the dominant phenotype and 1 being the recessive phenotype. So down here then, our phenotypic ratio is 3 tall to 1 short. And we predicted that based on this Punnett Square. Okay, so what does that look like in real life? So when Mendel did his experiments for his tall versus short, which is this part up here, he found that to be true. So his dominant plants, when he counted them, he had 787 tall plants. And his recessive short plants, he wound up with 277 short plants. So when we divide the dominant number by the recessive number, we get 2.84, which is very close to 3. And then, of course, our recessive over recessive is 1. So he applied this to multiple traits, looking at just one at a time, and he found essentially that all of those traits, when we had a heterozygous parent self-pollinated, produced 3-to-1 offspring every time. Okay, so that means that we had specific units of information being passed on in predictable mathematical ratios. So let's say that I have been given a tall pea plant as a gift, and I don't know what the genotype is of this pea plant. How can I find that out? So we perform a test cross. So because it's tall, it has the dominant phenotype, but the genotype could either be homozygous for tall or heterozygous for tall. So what we can do to find out which one it is we can cross it with a recessive phenotype, because the genotype for the recessive has to be 2 small t's. So in other words, it has to be a homozygous recessive plant. So if we cross-pollinate those two, then our two outcomes, or our two possibilities for our Punnett square, involve a plant when we have a dominant homozygous as a parental generation, then we are going to have this outcome here, which we've looked at before, and we can see that every one of them will be a tall plant. So if our parental plant that was gifted to me is a homozygous genotype, then we will have offspring that are all tall. Alternatively, if I have been given a tall plant that is heterozygous, when I cross it with our recessive homozygous, then I'm going to have half of my plants being tall and half of them being short. So a 1-to-1 ratio if my parental plant that was gifted to me is a heterozygous. So here I have made my cross. Here's my unknown gifted tall plant that was given to me, and I'm going to cross-pollinate that with a homozygous recessive. And it turns out that all of my plants are tall. All of the offspring plants are tall. So that means that my original plant had to be a homozygous dominant plant for tall. So why do we use a Punnett square at all? The answer to that question is because of our law of independent assortment. Remember, that means that there's equal chance for any allele to wind up in any of the gametes. So when we do our Punnett square, we put all the possibilities for the eggs and all the possibilities for the sperm. And in our monohybrid cross, that's pretty easy because you usually just have 2 choices for the eggs and 2 choices for the sperm. But the law of independent assortment is why we use the Punnett square. Okay, so in this scenario, we're looking at 2 genes. We're looking at a Y gene and an R gene. So the parent cell has 2 pair of homologous chromosomes that they are contributing. And during metaphase, all orientations of those chromosomes are possible, right? Because of the law of independent assortment. So if we look at that here, we have our blue chromosome lining up together with our red, or we could have them blue matching with red, blue matching with red. Okay, and then during metaphase 2, which would be down here, the daughter cell has only 1 member of each of the homologous pairs. Okay, so we either have red-red, blue-blue, blue-red, or red-blue. So that means at the very end of meiosis, these are all the combinations that were possible. And notice that our phenotypes are going to be based, of course, on the genotype. So we have dominant Y, dominant R, same thing. Oh, sorry. Is that right? No, I'm sorry. I meant not dominant Y. Recessive Y, dominant R, recessive Y, dominant R, dominant Y, recessive R, again, dominant Y, recessive R. We have both genes being dominant, Y and R, these two times, and then both recessive, YR, YR. So when we look at crosses that involve two traits, we call that a dihybrid cross. And so for each one, we have to denote the genotype. So for instance, our P generation here is tall and green, and this is homozygous dominant for both of those traits. And then we're crossing that with a short yellow P, so we denote that with homozygous short, which is lowercase t, and then homozygous recessive, which is yellow, which is denoted as lowercase g. So then we're going to cross those two, and we come up with a heterozygous tall and green plant. And in that case, the genotype is capital T, lowercase t, so heterozygous for tall, and capital G, lowercase g, which is heterozygous for green. So then we look at the F1 gametes and all the possibilities that could occur there. We could have dominant homozygous, dominant heterozygous for tall, and then we could have dominant heterozygous for green and dominant heterozygous for yellow. And that's both in the sperm and egg. And then we just have to cross every one of them, taking an allele from each of the sperm and the egg. Okay? And when we do that, we have our phenotypic ratio outcome, which is 9 to 3 to 3 to 1. And that's based on the phenotypic ratio. And so remember, when we have anything that has a heterozygous genotype, it's going to be a dominant phenotype. So in this case, it would be tall and green. So your allele key is here and what it means, and then there's the phenotypic ratio. And so that is a dihybrid cross. So human genetics follow the Mendelian model. And so here are just a few things, a few single traits that we can look at, whereas the dominant traits are freckles, the widow's peak that we've talked about, and a free earlobe. So do you have freckles or a widow's peak or possibly a free earlobe? The recessive traits are no freckles, a straight hairline, and attached earlobes. I actually have attached earlobes, so I guess that means my mom and dad both contributed recessive genes for me to have that. I do have freckles and I do have a widow's peak. Okay, so let's look at this just for freckles, for instance. So let's say that mom and dad are both heterozygous for freckles. So that means, let's say that mom up here contributed a dominant and a recessive allele. And then dad over here contributed, again, a dominant and a recessive. So what we would get out of that is a homozygous dominant, a heterozygous dominant, another heterozygous dominant, and a homozygous recessive. So that means we have a 1 to 2 to 1 genotypic ratio. But what is the phenotypic ratio? Remember, the phenotype is what it looks like. And for that, we have 1, 2, 3 that are dominant, so displaying freckles, and 1 that is not. So our phenotypic ratio is 3 to 1, just like we expected with the Mendelian genetics of the pea plants. Okay, so let's say then that mom has no freckles. So she is going to give us 2 egg cells. Oh, you know what I just realized? I did this backwards because this is a sperm, right? So, all right, let's say this is dad then. So sorry, dad. So dad, I guess, has no freckles, and we are adding 2 recessive sperm. And then mom then is going to add a dominant allele and a recessive allele. And when we look at the Punnett square for that, we have freckles, freckles, no freckles, and no freckles. So a 1-to-1 phenotypic ratio. So in other words, half of the offspring would have freckles and half would not. So the problem with human genetics is that you can't just do multiple crosses over and over and over again. First of all, our life generation is way too long. That would take years and years and years. And secondly, it's really not ethical to say, let's see how many we can produce out of you. So instead we do what's called a pedigree analysis. So that's looking at your family tree and tracing a particular trait. So let's say, for instance, we're going to use this as our key. The women are circles and the men are squares. And the circle is going to be a woman that does not have the trait, so an empty circle, no trait. And the filled in circle does have the trait. Same thing with the man. If the square is empty, he does not have the trait. If the square is full, he does have the trait. Okay, so let's look at the family pedigree of this lovely family. We're going to call them the Hartmans. And we are going to look at a trait that deals with the heart muscle. And in the dominant form, they produce a healthy heart muscle. In the recessive form, there's abnormal heart muscles that cause problems. So in order to have that heart defect, you have to have a homozygous recessive trait for this. So when we start, we know, hang on, sorry, I'm trying to get my pen. So we know that Joshua and Abigail right here have heterozygous alleles for this heart muscle gene. For John and Hepzibah, which is a lovely name, you know she's pretty, we know that they don't display the abnormality. So we know they have at least one dominant allele, and we don't know about the others. So Joshua and Abigail have 2 children. One of them has the heart defect. So we know that Jonathan has a homozygous recessive genetic. We don't know about the other child. We just know that she does not have the heart defect. So John marries somebody named Elizabeth, and Elizabeth is heterozygous for the trait. And how do we know that? We know that her parents did not have the defect, so she has at least one of the dominant genes, one allele. But when she and Jonathan have children, some of them show up with the defect. So that means she had to at least contribute one allele of the recessive trait. So that's how we can trace the family pedigree when we're looking at our family tree. And so here are all the children. So now that we know Jonathan and Elizabeth's alleles, then we can trace out the rest of their children and know which ones are heterozygous and homozygous. And we know that the ones that don't have the defect have to be heterozygous because Jonathan was homozygous, so all he can contribute is a recessive allele. So that means that each of these children have at least one homozygous recessive allele. So that brings us to this point with Jonathan and Elizabeth's children. Since all of them have at least one recessive allele for the heart defect, they are what we call carriers. So that means that they have a normal phenotype because they still have one of the dominant alleles, but because of this recessive allele, they are a carrier. That means that they can pass it down to their children. So a carrier is a heterozygous person who carries a disease allele. So one of Jonathan and Elizabeth's children, named Charlie, is going to come in with being a carrier, and he is going to then marry somebody named Angie, and they are going to have children. And if Angie is also a carrier, then what we have then is our normal Punnett square with a 3-to-1 ratio, which means that 75% of their children are likely to have a normal heart, whereas 25% of their children, or a 25% chance that if they have a child, it will have a heart defect. Okay, so with Charlie and Angie's offspring, we said that there's a 75% chance that they will have a normal child, and a 25% chance that they will have a child with a heart defect. But in addition to that, when we look at this chart, we also note that there's a 50% chance that their child will be normal but will be a carrier also. Cystic fibrosis is one of those examples of an autosomal recessive disease. So in cystic fibrosis, there is a particular channel protein for chlorine that is defective. And so those who have a homozygous recessive C allele then will have cystic fibrosis. So when we look at autosomal recessive pedigree charts, this is what it's going to look like. And you can tell this person right here would be affected by the disorder. Somebody like this is going to be a carrier. And again, here we go, 2 that are affected, and we know that it's an autosomal disease because it's male and female, so it doesn't matter who the sex of the carrier. And so that is how we look at that mapping of the autosomal recessive disease. Okay, so what about dominant autosomal diseases, where the dominant trait means that you're expressing the disease whether you are homozygous or heterozygous, and the only way to escape that genetic disease is by being a homozygous recessive. Well, here are just a couple of examples of that. We have Charge syndrome, Marfan syndrome, hypercholesterolemia, sorry, I can't say that very well. And this one in particular is quite fascinating. Polydactyly. This is having an extra finger or toes, and it's actually fairly common in humans, dogs, and cats. So those of us who have 5 fingers and 5 toes actually have a homozygous recessive trait. Let that sink in for a minute. So here we go. If we are looking at our Sam and Susie, and they are both heterozygous, if they are heterozygous for the disease that is extra digits, that means that 75% chance that their children will have extra digits, and only 25% chance that they will be normal, in quotes, that only have 5 fingers and 5 toes. So far everything we've talked about has been pretty straightforward, genetically speaking. So let's just real quickly go through some variations on Mendelian genetics, because it is a little more complicated than just straight dominance and recessive, for some genes anyway. So one of the variations is what we call incomplete dominance. That means the phenotype is a mixture of the dominant and recessive phenotypes. If you'll remember, this goes back to the common thought of the day before Mendel even came up with his theory of the particulate inheritance. So this means that, for instance, our dark brown horse here, the dominant gene is the capital B, and so if we have 2 of those, we're going to have a dark brown horse. And then by nature, the recessive trait is a white horse, so lowercase b, lowercase b. But when we have a heterozygous, capital B, lowercase b, we wind up with a tan horse. So incomplete dominance, a mixture of the 2. And we see this phenotype in plants as well. So if you've ever seen pink carnations, that is because of the crossing between a red carnation and a white carnation gives us incomplete dominance, so we get a pink carnation. Interestingly enough, we can also have what we call codominance, which means both of the alleles are expressed at the same time. That's usually a bit rarer, but in that case, we would have a red and a white being the dominant and recessive, and then when they're both expressed, we have a red and white petals both on the same flower. So that's codominance. Another example is the brindle pattern in dogs. That is a codominance pattern. Now another variation is what we call multiple allelism. So instead of having just your typical 2 alleles for each gene, there are multiple versions, and our red blood cells are great examples of that. So we have 3 different allele types, an A, a B, and an O. And so red blood cells are identified as A, B, and O based on the glycoproteins on the outside of the cell. And so I don't know if you know your blood type. My blood type is A, and so I am right here. And each person has 2 different alleles. But there are 3 alleles total as options for your blood type. So that is called multiple allelism. And then there's polygenic inheritance, and this is one that I find fascinating. And it's one trait that is the result of an additive effect of more than one gene. And so skin color is a great example of this. So how many genes are responsible for skin color? Many, many, many. And they all contribute to give a spectrum of skin color. I think it's fascinating. Here are some other traits that require multiple genes, so other polygenic traits. Not only our skin color, but our hair color, our height and weight, our eye color. So, for instance, I have very dark brown eyes, and I have one daughter who has super dark brown eyes. That's Claire. But my oldest daughter has very light brown eyes. And then I actually also have a daughter with very light blue eyes. Yes, my mother has blue eyes, and my husband's mother had blue eyes. But it is a polygenic trait, so it's more than just one gene. And so I like to think of it almost like dosage, like how many of those genes are turned on determines how much pigment you get in those in either your eyes or your hair or your skin. So I tend to call that dosage dependent, but it's technically called polygenic. Okay, so chromosomal inheritance. What we've been talking about so far has all been a part of our autosomal chromosomes. And most of our chromosomes are autosomal, right? 22 pairs of them. But there's one pair that are sex chromosomes. And so through meiosis, we wind up with half the DNA. So here we go with our 23 and our 23 and our 23, and these are all our sperm cells right here. And so when we look at our male germ cell, we have X and Y chromosomes, whereas our female germ cell is going to have just the X chromosomes. So when we divide those by meiosis, in our males, we'll have, percentage-wise, two female carrying, two sperm carrying X chromosomes and two sperm carrying Y chromosomes, whereas all the egg cells are going to carry just the X chromosomes. Okay, so the point is that the sperm determines the sex of the offspring, and you have a 50-50 chance of either having a boy or a girl. Now I'm just going to throw a caveat in there just to let you know that there was a study in the early 2000s that determined that the egg cells kind of had a say-so in which sperm cells they let in, chemically speaking, of course. So in that sense, yes, the sperm cell does determine whether you're going to wind up with a male or female offspring, but there is some selectivity based on the egg cells. But for now, let's just say we cross, like we normally do, our X and Y and our X and X. So in this, then, we would have the possibility of girls right here, two of those, and then two possible combinations for males. So this is where we get our 50-50 or 1-to-1 ratio of male versus female offspring. Okay, so then besides just determining the gender of the offspring, what types of genes are on these chromosomes? On the X chromosome, obviously is genes for being female, but also genes for everyday human function. And the Y chromosome actually carries very little genes, but just genes for being male. So the development of an embryo into a male. So because genes for everyday function that are on the X chromosome can carry diseases with them, the genes on the X chromosome are called X-linked or sometimes called sex-linked. So here are some examples of the genes on the X chromosome. One is a gene for blood clotting protein. When we carry the recessive, that results in a condition called hemophilia. So the inability to clot your blood. Secondly, there's a gene for a muscle protein called that results in a condition called Duchenne's muscular dystrophy. And again, that's a recessive condition. And thirdly, and probably the most fun really, is a gene for an eye pigment protein. And the recessive condition of this one causes colorblindness. So when people have muscular dystrophy, that means that in their muscle tissue, if they carry this abnormal gene, it means that they start to begin to have fibrous tissue within their muscles. And that makes them stiff and unable to be used as well. So they're not as flexible like you would expect your muscle tissues to be. So we actually have a good friend whose son has this, was in my oldest daughter's playgroup, and they didn't really notice until he hit about 2 years old, because that's when you start doing a lot of your muscle development and developing your motor skills, and that's when it becomes evident. So the way we denote genes that are sex-linked is if it is a dominant allele, we would put the X with a capital A, that indicates that it is sex-linked on the X chromosome. If it is a recessive allele, then we have the X with the lowercase a. So obviously for a woman, if they are dominant for any of these diseases that we talked about, because these particular diseases only appear when you have a homozygous recessive gene, then if it is a woman that carries a homozygous dominant for these, then we say no disease here. We can have a sex-linked heterozygous genotype, which means you have no disease but you're a carrier. And then the sex-linked homozygous recessive, of course, means that it's a woman with a disease. Now, if we're looking at a man, then we have to include the X and Y chromosome, and we only have the genotype written by the X chromosome because it's X-linked. And so a man with a dominant sex-linked gene would have no disease. A man with a recessive sex-linked gene would have the disease. So let's talk about colorblindness for a minute, because that's just a quick, easy, genetically inherited, quote, disease. And so if we're looking at a woman, a man, that's not what I meant to say, a man because he has a Y chromosome, but let's say this man is dominant, and that means that he has normal vision, and then we're crossing that man with a woman who is a carrier for the colorblindness gene. So when we do the Punnett Square for their offspring, here we have a woman with no color blindness, another woman with no color blindness, a man with no color blindness, and a man with color blindness. So our percentages then mean that we have a 25% chance of having a child that is colorblind, right? Because only 1 out of 4 is colorblind. The percentage of having a boy that is colorblind is 50%. And the reason we say 50% is because we have 2 boys and 1 out of the 2 have colorblindness. The percentage of having a girl that is colorblind is 0% because we have 2 girls, 0 of which have colorblindness. So here is your red-green colorblindness chart. So you should be able to look at this image, and if you have colorblindness, you will not be able to see the number that is present. If you are not colorblind, you will be able to see the green number. And so I'd be interested to see which of you are colorblind and which of you are not. So please send me an email, and in the subject say colorblind-yes or colorblind-no. And then we'll get together a chart from our particular class of how many of us are colorblind and how many of us are not.